Well, welcome back and welcome to starting our studies in our option. So we are going to be looking at medicinal chemistry for our option, and let's jump straight into it. So the first topic that we're going to look at is actually introducing pharmaceuticals, what it means when we say pharmaceutical or drug, and having a look at some of the expectations for our assessment. So our option is broken down into 6 topics, D1 through 6, at least for SL, and then there's a couple of extra topics for HL that we will look at. The first one that we're going to look at is this one here. Medicines and drugs have a variety of different effects on the functioning of the body. So let's get going. One of the first things that we have to do is to define what it means when we're talking about drugs, medicines, pharmaceuticals, or different things. In society, we tend to, when we talk about drugs, it tends to have a negative connotation. We tend to be talking about things like LSD or methamphetamine or ice or things like that. It's often the term that's used when we're talking about drugs that don't seem to have any particular therapeutic index. In terms of definition, though, they are the same. So the general definition of a drug or a medicine is any chemical that does one or more of the following things. It can alter incoming sensory sensations, so that can be making someone feel high or woozy, dissociated, giggly, those kinds of things, make things seem slower, faster, have visual effects, those kinds of things. It can alter our mood or emotions, so by making us feel happy, sad, no inhibitions, those kinds of things. It can alter the physiological state, so this can be consciousness, so knocking someone out, making them sleepy, perking someone up. The activity levels, such as stimulants and caffeine and things like that, or change the physical condition, which is where we get the ones that are associated with repairing the body or curing disease. So if we look at something like the inflammation response, which you may have done in biology, okay, we need to think about what does something like a painkiller, otherwise known as an analgesic, how does it alter the state of the body? Okay, in this case, it's going to change some sensory sensations, such as stopping pain. Okay, so where you were in pain before, it's going to stop that pain or stop that message from getting to your brain. It's going to reduce inflammation, so usually it reduces swelling, reduces inflammation, and can reduce fever, those kinds of things. So we take drugs or medicines for a large number of reasons, but largely this is the definition that we need to concentrate on, that a drug is anything that alters the sensory sensation, emotion, or physiological state of a patient, including condition, mood, or otherwise. So this is what we can consider a drug or a medicine. The term is used interchangeably. So where do our drugs come from? Largely, drugs are sourced from a wide variety of different things. Most of them historically have been done from natural products. There are very few drugs which have actually been designed from scratch without having something that sort of indicated they had a pharmaceutical purpose to begin with. So a lot of them come from plants. So poppy flower gives us morphine. This tree here gives us another drug here, which I've forgotten the name of briefly. Taxol we will study a lot in quite detailed. It's a cancer drug that has been pulled from the yew tree. This sponge here and the arrowhead viper, this one here, the viper, is actually a heart drug. It looks at changing blood pressure and things like that. And if you think about a lot of poisons and things, they change blood pressure to incapacitate the victim. And this is quite useful for things like heart disease and other things like that. You'll notice a lot of them have quite complex structures. This is common for drugs with natural products, these kinds of bridging groups and things which are quite challenging to make within a laboratory synthetically. So a lot of the time, a lot of the drugs that we use are still extracted from their natural product, or at least the precursors of them are. So we do need to know our drug development process, and essentially what this means is how do we go about manufacturing a drug from start to finish? And this is quite a complicated process. In essence, for every 25,000 compounds that start their way into being a drug, only about one or two of them will get to market. So it costs a lot of money. There is a lot of failure along the way, and it's quite a rigorous process. What this means is a lot of that money needs to be remade when the drug gets to market. So they can be quite expensive, and we need to regulate very carefully how these drugs get onto the market. So the release of a new drug is very strongly controlled by government authorities, and it's different in every country around the world. The specific regulations that they have are very different, but the process is much the same. Finding a lead compound, okay, so a lead compound, which is going to start your drug development process. And that starts first by knowing where you want the drug to go. So we have this one here, which is target selection. So this is defining where in the body do you want it to act? What are you trying to cure? Is there a gene sequence? Is there a particular cellular process? This is the biological aspect of drug design, largely. Finding proteins that are involved in a disease process, or understanding the physiology of that disease and how it might be treated. Then they have lead discovery. So lead discovery is like a lead compound. This might be testing thousands of compounds against your target and seeing if anything has any response at all. They use different methods, synthesis or isolation from a natural product, combinatorial chemistry, a lot of robots so they can process thousands of screens quickly, okay, because remember, out of 25,000 compounds, we're probably only going to get one drug. From that, it goes through the medicinal chemistry arm. So once there's a lead compound, medicinal chemistry looks at how are we going to synthesize it? Can we synthesize it in ways that make it more effective? Can we use computational chemistry, which is what we refer to as in silico chemistry, to improve that? And can we make it efficiently in high enough yield that it would be valid? From there, we go into in vitro studies, which is glass. So we look at cell assays, so not animals at this stage, just cells, different cell lines that are grown up, look at its mechanism of action, how is it working, is it effective? From there, the unfortunate thing, I guess, about pharmaceutical development is we still use a lot of animal models and disease states and things like that, imaging. So we start to go in vivo into the body, usually first up in mice and then gradually into other animals as well. From there, after it's passed any in vivo studies, it gets to clinical trials where we start to trial it on humans. So this process, as you can imagine, is very risky because your drug may get as far as a clinical trial and then have to go back to the drawing board. It may fail, it may fall down. So we're talking, between 5 to 10,000 screened. Some say 25,000 screened. We'll get one drug out the other side. This can cost hundreds of millions of dollars and generally takes anywhere around 10 to 15 years, not much less. You won't get a drug onto the market quickly. Okay, so discovery and preclinical testing can take 4 to 6 years. Okay, and then the human trial aspect of it will actually take another 8 years or more trying to figure out how the drug works. Does it work in humans? Is it safe? Are there long-term side effects? All those kinds of things. So we have identification and selection of the disease, identification of the pharmacological target, so is it an enzyme, protein, or other thing like that. We identify the lead molecule. We go into preclinical trials, okay, which is usually in animals and things like that. Clinical trials, reporting of those results, which are then looked over very carefully, monitoring of the drug after the market release, because sometimes a drug needs to be pulled off the market after it's been given approval. Okay, so that involves a lot of patient recruitment, constant following of data, preparing reports, and following to see if this will actually work and is it going to be safe. So for our preclinical trials, there are two types. There are in vitro and in vivo testing. In vitro testing is testing on cells and tissues that aren't actual animals, and we look at the efficacy, concentration, cellular uptake, work out what dosages are required and those kinds of things initially. We look at toxicity. Okay, so how is it toxic? Why is it toxic? Can that be minimized? We look at potency and things like this. Once all the questions about how it's work, where its toxic levels are, then it can be moved on into in vivo testing. Okay, so we go into animal testings, which differ from country to country, and is a very debatable ethical issue in terms of the pharmaceutical industry. Is it right to be using animals? When is it wrong? What are the risks associated with that? Once this happens, we test in live animals to establish an ED50, which is an effective dose in 50%, and an LD50, which is a lethal dose in 50% of the population. Okay, so we will talk about those a bit more. As you can imagine, many drugs never even make it this far. Okay, so we test in 3 different animal models, usually mice, then some sort of dog, often beagles actually, and into chimpanzees, primates. Sometimes pigs are used also because of their similarity in organ structure. So for that, we look at all the possibilities of where this can go wrong. Once the clinical trials are done, we start to, preclinical trials are done, we move on to clinical trials. This tests them for their effectiveness in humans. Sometimes they're effective in smaller animals, but they're not effective in us at all. Sometimes they're non-toxic to smaller animals, but are toxic to humans. So this checks the effectiveness, looks at the therapeutic window, which is how we measure the safety of a drug. The tolerance, does a tolerance build up quickly and therefore the drug become ineffective? Side effects, placebo effect, all these kinds of things are checked. It's a blind trial, so half of the people will get the given drug, the other half will get a placebo, and then the results are checked for validity doing that. Once this stage of the clinical trial is done, you'll often see structural modifications being made, ways to reduce side effects or perhaps improve solubility, get more of the drug effectively to where it needs to be. But these are all small modifications. Any major modifications, the drug needs to go straight back to the drawing board and start the process all over again. So discovery, 2 to 5 years, preclinical testing, 1 to 2 years, and then 6 to 10 years of clinical trials in humans before the drug will even make it to market. So this is basically a quote from the safety report on drugs, and it states that no drug that is pharmacologically active is entirely without hazard. The hazard may be insignificant or may be acceptable in relation to the drug's therapeutic action. Furthermore, not all hazards can be known before a drug is marketed. This is basically an understanding that side effects differ from person to person because our biological makeup is different, but also anything that has a pharmacologically active response in our body, when taken in extreme, can cause problems. Okay, everything we take that has an effect on our body can have a good or bad effect, and this needs to be considered when we put a drug onto the market. Okay, and then of course there are situations when regulations fail, so I highly encourage you to read the article about the drug trial in France where a young man died, and we'll also of course be looking at thalidomide in quite a bit of detail, and we'll do that in class. So that is your introduction into how drugs are discovered.